Hello and welcome everyone to this video podcast for Evolution and Diversity. We've completed the first unit of the material, and in that unit we focus pretty heavily on evolution. And in this unit we're going to start by taking that next step from evolution and talk about speciation. How are new species formed and how do we classify those new species? So we're going to begin by talking about the evolutionary organization of living systems, and then we're going to move into criteria that can be used to identify a species. We'll learn that there are 3 different ways that we can categorize a species. And then we'll finish by talking about reproductive isolating mechanisms. Okay, let's go ahead and get started with our first objective. Okay, let's go ahead and begin our discussion of evolutionary organization of living systems. As we get started with this discussion, I want to think back to our last unit of material, when we talked about Darwin's theory of evolution by natural selection. This theory, remember, made 2 predictions about the nature of species. The first, remember, was that species change through time. And the second is that species are related by common ancestry. So thinking about these 2 predictions, it seems that we should be able to come up with some sort of tree of life. That is, some kind of explanation as to how all organisms are related to each other. And for that to be the case, there should be one common ancestor for all of life. So one way to think of a tree of life is the phylogenetic tree that includes all organisms. And so I introduced a new word here, phylogenetic. So let's define phylogeny. And this is the evolutionary history of a group of organisms. And we're going to come back to this word phylogeny a little bit later today and also throughout the semester. So to think about this tree of life and phylogeny, let's look at a phylogenetic tree here that shows all the domains of life. So let's draw this tree of life in a fairly simplistic manner at first. So down here, at the very bottom of this phylogenetic tree here, is the common ancestor of all of life. So as life evolved from this common ancestor, there were several places where changes occurred that led to new species. And whenever there is a split in this phylogenetic tree here, we call it a node. So this would be called a node. And a node represents the common ancestor of all of the species that come off of this line that I'm showing here in orange. And all of these species that come off of this node here are called bacteria. And from this, we know there are many other types of bacteria that are also represented in this domain of bacteria. Now, this second domain that branched off the tree of life here came from this node, and all the species that evolved after it broke off here fall into the domain of archaea. And also remember that there are many different species in this domain of archaea. And we'll talk about some of these later on. But they all have this one shared common ancestor right here at this red circle, this node. And then the third domain of life are the eukaryotes. Eukaryotes, of course, also have several different species that will branch off of it as well. But they're all eukaryotes. So the big take-homes from this, I'd like you to remember, is that all of life exists in these 3 domains, bacteria, archaea, and eukaryotes. We can represent all of life on this phylogenetic tree that shows the evolutionary history of all the organisms of life. We can tell a little bit about the history of them because we know that all the species that branched off to form archaea from this node have a certain set of characteristics in common. Same thing here. All the species that broke off from this node to form bacterial species all have a set of characteristics in common that organizes them into this domain of bacteria. Same thing with eukaryotes. All the species found within the domain of eukaryotes have certain traits in common. And also remember that all of life, bacteria, archaea, and eukaryotes, can all be traced back to one common ancestor. Now, we may not know what this was, and it certainly doesn't exist today, but there was a common ancestor that led to all of the life that we see now, based upon the predictions of natural selection. I also think it's important for us to remember that This phylogenetic tree here of all of life, it's a work in progress. As we learn more about all the different life on Earth and all the different species in these different domains, sometimes we have to make changes in what we know about their evolutionary history. And that's just the process of science. As we looked at that previous whiteboard with all the different domains, I want to say something about this word called taxonomy. This is the effort to name and classify all organisms. So every organism that we find, we want to place it within that phylogenetic tree, within the correct domain, within the correct phylum, within the correct families. And I want to go over specifically one of those classification systems, and that's how we classify a species. So there is a system for naming species. So every species has two names, and we call this binomial nomenclature. The first word of a species name is its genus. And a genus is a group of closely related species. And then the second word in the name of a species is the species name. So for instance, humans are Homo sapiens. So within the genus of Homo, there are many similar species, of which all except Homo sapiens are extinct, but there are Homo erectus, Homo habilis, and others. And then sapiens is to specifically name the species that we're talking about, humans. Just to give you another example, a fruit fly is Drosophila melanogaster. And one of my favorites, a typical budding yeast, would be Saccharomyces cerevisiae. So these are just three of many examples, because every species has this binomial nomenclature. A few things I want to point out here that you may or may not have noticed while I was writing these out is that when we write these out, the first word, the genus, is always capitalized. The species name is always lowercase. The names of a genus and species are Latin, and since they're Latin, they have to be italicized. And if you're writing it out, it's hard to write in italics. I know I certainly couldn't. So we always represent italics in our handwriting as an underlined word. So if you're typing it, you would use italics to represent a genus and species. If you're handwriting it, you would underline it. But still, always the genus is capitalized and the species name is not capitalized. Okay, let's go ahead and move on to our second objective. All right, so let's talk about this second objective for today, and that includes the criteria used to identify species. And as we begin this discussion, let's give a working definition of species. One way we can look at a species is as an evolutionary independent population or group of populations. We can also think of a species as a group of individuals that actually or potentially could interbreed in nature. And then the next term I'd like to define is the term of speciation. Speciation is the splitting event that creates two or more distinct species from an ancestral species. So essentially, how is it that we create new species? And these would be speciation events. So now let's spend a little time talking about how we would be able to determine if what we're looking at is a separate species, or should it be included with another species. So there are three different species concepts. The first is Morphle Species. The second is Phylogenetic Species. And the third is Biological Species. All three of these concepts are useful in trying to determine if something is actually a different species. However, each of these also has some disadvantages associated with them. So what I want to do is spend a little bit of time talking about each of these, as well as talking about the advantages and disadvantages of each. So let's go ahead and start talking about morphle species. And another way we could say this is morphological species. Morphle species works around the principle that distinguishing features are most likely to arise if populations are independent and isolated from gene flow. For instance, if you had a large landmass here, maybe a continent of some sort, and you had bird species here that had large beaks, and then on an island over here, maybe you had bird species that had small beaks. Other than that, these two groups of birds might have been pretty similar looking, but when you look at their beaks, they're quite different. So that is a distinguishing feature between these two populations of birds. Now, if these two populations are independent, which they are, they're in different locations, and they're isolated from gene flow, meaning the birds on this island and the birds over here are not going to trade genes from each other, because we will assume that these two populations are far enough away from each other that they would not normally interact with each other. And if that's the case, then a reasonable prediction might be that these are actually different species and they are not the same species. Now, some of the advantages to identifying a species this way is that it's widely applicable. We can use this method of identifying species with sexually reproducing organisms, asexually reproducing organisms, and also from the fossil record. So it doesn't have any limitations there. So some disadvantages of identifying species using morphle species include it could lead to errors in polymorphic species. A polymorphic species is a single species, but they look different within that species. So maybe you have cat populations where some of the cats are spotted and some of them are a solid black color or a solid brown color. Turns out they're the same species, but morphologically speaking, they have different colors. So someone might have come along and said they're different species. And the other disadvantage I want to point out is when we have cryptic species. Cryptic species are species that are actually different species, but their differences are more than just morphology. So they may look very similar. You might have two bird populations and they look very similar, but when you start looking more closely, you might discover that, for instance, they have a different song. They sing a different song, and that may lead to them being different species. And I should mention this third disadvantage, and that is that sometimes the way we view morphological differences can be subjective. One scientist might view a morphological difference between these two populations, but another scientist might look at those same two different populations and not think that the morphological differences are distinct. Okay, let's go ahead and move on to the phylogenetic species concept. This method identifies species based on evolutionary history of populations. In this way of identifying a species, we think of a species as being the smallest monophyletic group on a phylogenetic tree. So I think we need to define what a monophyletic group is. So a monophyletic group consists of an ancestral population and all its descendants, and only those descendants. I think the best way to see what I mean by a monophyletic group is to look at a phylogenetic tree. So let me draw one of those now, and then I think that will serve as a good tool to understand how we use these phylogenetic trees to identify species. So here's a phylogenetic tree, and we can think of a monophyletic group as anything past a certain point. So that's kind of vague. Let me tell you what I mean by that. If we were to cut this tree right there, then the monophyletic group would be everything here, but it would not be anything down here. So let me just draw a circle around this. So this would be the monophyletic group. It includes this ancestral population down here, plus all of its descendants. However, the way I cut this here is it includes different species, not just one species. So let's erase this and recut this. One thing I should have said when we started this is at the end of each of these lines is a different species. And so a species is defined as the smallest monophyletic group. So again, if you cut it here, that's not the smallest. But if you cut it here, then this becomes a monophyletic group in itself. And that's the smallest. You can't get any smaller than that, because remember, when we cut it here, it included all of this, right? If we cut it here, it would include everything we see here. But this time, we cut it right here, and we only see this one species identified here. I haven't written the name of the species yet, but I will in a moment. Based upon the phylogenetic species concept, the smallest monophyletic group is a species. This would be a species, 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 and our good friend up here would also be a species. Now, I used the evolution of bears as an example, because who doesn't love a bear? So here we have polar bears. Here we have brown bears. Here we have North American black bears. Here we have the sloth bears, sun bear, and the Asiatic black bear. If you happen to be a fan of the speckled bear, I couldn't include this here because I just ran out of room. It's not an insult to speckled bears or the fans of speckled bears. It just didn't fit on here. So let's summarize what I'm talking about here. The phylogenetic species concept is based upon looking at monophyletic groups. Not just any monophyletic groups, but the smallest monophyletic group here. That identifies a specific species. Again, as I said, if we cut it here, then yes, as I drew before, all of these three species would be in that monophyletic group. But we don't want to do that because we only want to consider the smallest monophyletic group, what I have squared in here at the ends of each of these lines. Those are the species. There's one other term I want to define for you, and that is synapomorphy. So these are traits found in certain groups of organisms and their common ancestors, but is missing from distant ancestors. For instance, we would say there is a synapomorphy that occurred here that led to the polar bears, and this synapomorphy would be white fur. It distinguishes polar bears from all the other bears, and we would see it, of course, in polar bears and all of their ancestors on this line. We haven't specifically talked in great detail about phylogenetic trees, but this line represents many different species that eventually led to the polar bears that we see today. But many of these species here, of course, are no longer around. They have gone extinct, and all we're left with are the polar bears. But all of those ancestors all would have had white fur, but none of the distant ancestors, anything, say, to the left of this line, so that would be anything here, it would include the brown bears, the black bears, sloth, sun, and Asiatic black bears. None of them would have had the white fur. That is unique to this polar bears. That is unique to polar bears. And a way that we often can identify synapomorphies is by looking at DNA sequences. So let's just write that up here. Building phylogenetic trees like this can be more specific if we know the DNA sequences, and then it's much easier to identify where changes have occurred within different species within the phylogenetic tree. So there would have been a mutation that led to a population that ultimately became polar bears, and that mutation represented at this synapomorphy is not found in brown bears, is not found in black bears, sloth, sun, or Asiatic black bears. So this approach has some advantages, and those advantages include what we saw with the Morphle Species examples, and that is that we can use this to identify sexually reproducing species, asexual reproducing species, and species that we can identify in the fossil record. Similarly, as we saw with the Morphle Species, any one of these species, the only way that they're going to have different synapomorphies is if they are isolated and independent from the other species. That is, no gene flow between them. There are, of course, some disadvantages to this approach. These very specific phylogenies like this don't exist for all species. We simply haven't had enough time and enough data to build them all. And so while they're growing in numbers and we're able to use more and more of them, the overall numbers of them might not be enough to identify all the species we need to identify. We'll get there, we're just not there yet. Another disadvantage, and I don't know if this is really a disadvantage, it's just the way it is, this approach will identify very small differences between populations in their DNA sequences. And so it might include more different species than the other concepts. It would be surprising if the phylogenetic approach identified, say, 2, 3, 4, 5 times more species within certain areas compared to the other ways we identify species. That may not be a bad thing, but it is a reality. Okay, let's move on to our last method of identifying species. All right, now let's talk about this third concept of species, and that's the biological concept. And this is best described as reproductive isolation between populations that don't breed and don't produce viable and fertile offspring. Now, there are many different ways that populations could be isolated from each other that prevent them from breeding with each other. And if they did try to breed with each other, they would not produce viable offspring or offspring that could reproduce on their own. And I'm going to go into those details in just a little bit. But what I'd like to do now is finish off this by talking about the advantages and disadvantages of biological species concepts. The advantage is that reproductive isolation equals evolutionary independence. If different populations are isolated from each other in a reproductive way, then that allows that population to evolve into a new species because there's no gene flow within it because it's isolated from other populations in its ability to reproduce with them. All right, the one disadvantage I want to point out here is that you can't really use this concept of biological species if the organism is asexual. So it's not useful for identifying asexual species. Many species reproduce in an asexual manner, so if they're reproductively isolated, it doesn't matter. This way of identifying these asexual species is well, rather useless. So now let's use these three concepts in an example, and let's use it in an example to determine how many species of elephants are there. When I was in school, I learned that there were two species. There was the African elephant and the Asian elephant. The African elephant had large ears. That was an easy way to distinguish them. And the Asian elephant had small ears. What has happened over the years is that scientists have discovered that within Africa, there are two populations of elephants. So within the African elephants, we have two populations, as I was saying. The smaller forest elephants found in Central Africa, and the larger elephants found in the savanna. So these two populations of elephants found in Africa raises the question, are there two species of elephants or are there three species of elephants? And so let's apply the three concepts to see if we can figure out if there are two or three species of elephants. So let's first consider the Morphle Species concept. When they look at the morphology of these two elephants in Africa, they see different sizes. And when I say different sizes, I mean their overall size is different, their tusk size are different, their head sizes are different. That forest elephant is just smaller compared to the savanna elephant. So just looking at the morphological differences, one might say that there are three species. Because if you have different morphologies, then there's a good chance that those were independent and isolated populations. Well, let's now look at the biological concept. Okay, so let's think about the biological concept here. Remember, this concept is centered around whether or not these two different elephant populations breed with each other and produce viable and fertile offspring. Well, one thing to consider is that they live in different habitats. One lives in the forests in Central Africa and the other lives in the savanna in Southern Africa. They don't really interact with each other. You wouldn't expect them to interact with each other. And since they don't interact with each other, one might assume that they are reproductively isolated. However, in captivity, they do breed and produce viable and fertile offspring. Now, how successful this is a little unclear, but the bottom line is that they could actually produce progeny. This might lead you to believe that there are two species. But it's not really definitive, because in their natural habitats, they would never come in contact with each other. So it's a little inconclusive looking at the biological concept data. All right, well, let's see the phylogenetic concept data will shed a little bit more light on that. And so we're going to need to erase these two to move into that. All right, so let's look at phylogenetic data here. So now, with the phylogenetic concept, the key is you have to make a phylogenetic tree, and that's what they did. And I'm going to show you what they found in looking at all three populations of elephants. Here you see the Asian elephants. Here you see the African forest elephants. And down here you see the African savanna elephants. And to build this tree, what they did was they took tissue samples from all of these elephants and sequenced their DNA. And they were able to find unique sequences, differences in all three. So they all three had evidence of synapomorphies based upon their DNA evidence. So at the phylogenetic level, looking at their DNA sequences, the Asian elephant, the African forest elephant, and the African savanna elephant are all three different species. And so this is pretty definitive proof that what we had always considered to be two species of elephant is in reality three species of elephant. And if you remember at the very beginning of today's material, I said something on the lines of our tree of life that we know is a work in progress. We're constantly evaluating it and reevaluating it to see if new information, new data can change the way we used to think. That's what science does. We had more data, we applied it to what we thought, and it turned out that what we thought before was not accurate. The new data now tells us we have three species of elephants. That will no doubt be a great conversation starter when you go home and visit with family. You can tell them that there are three species of elephants and not just two. All right, now it's time to move on to our third and final objective for today's material, and that is to talk about reproductive isolating mechanisms. And these are the reproductive isolating mechanisms that are used when we think about the biological concept of species. Because remember, in that concept, it's all about whether or not two populations are reproductively isolated. And when I mentioned that before, I said we'd come back to that because there are different ways that populations can be reproductively isolated. And we can put these into two large categories. The first is pre-zygotic isolation. And the second is post-zygotic isolation. So where do these words come from and what do they mean? Remember that fertilized egg is called a zygote. So pre-zygotic isolation is any reproductive mechanism that prevents the formation of a zygote, that prevents that formation of that very first embryo. Post-zygotic isolation involves any mechanism that allows for fertilization to occur and to form that zygote, but that zygote does not form a viable progeny. And there are 6 ways that we can have pre-zygotic isolation. They include temporal, habitat, like we saw with the elephants on the last whiteboard, behavioral, mechanical, and gametic barrier. There are 2 ways that we can think about a post-zygotic isolation. One is hybrid inviability, and the second is hybrid sterility. So what I'd like to do now is to take each of these examples and define what they are and then provide an example of each of them. So temporal refers to a time period. So this type of reproductive isolating mechanism means that the populations have different breeding seasons. So you may have an area that contains many different species of frogs. But the different frog species will have different times of the year where they become fertile and when they breed. And because of that, the two different species will never breed with each other just because they have different breeding seasons. The next one we want to talk about is habitat. And this is when breeding occurs in different habitats, in different areas. A good example of this would be the Arctic fox and the gray fox. Very similar in many other ways, but the Arctic fox lives in the Arctic regions above Canada, and the gray fox is found in southern populations, mainly in the United States. So their habitats are different. They never come in contact with each other. The next one is behavioral. This is often seen when the two different species have different courtship behaviors. A good example of this would be birds. You could have different species of birds living in the same area, the same forest, wherever they might be, but if they have different song patterns, they're going to attract only members of their species. They won't attract members of different species. So they have a different courtship behavior. And so that allows them to be reproductively isolated. Mechanical is the next one we should talk about. And this is when reproductive structures are not compatible. Good examples of this are many plants. The reproductive structures might not be able to accept pollen from other species of plants. Snails is another good example. Different species of snails have reproductive structures that are also not compatible. Okay, the last one for prezygotic is gametic isolation. And this is when the sperm and egg are incompatible. The sperm and egg do come in contact, but because they're different species, they cannot fertilize each other. And because of that, these two species are reproductively isolated. Examples of this include sea urchins. Different species of sea urchins will often be reproductively isolated because their sperm and eggs are incompatible. We also see this in many plant species as well. Okay, let's talk about post-zygotic mechanisms. And remember, there were two of those. The first is hybrid viability. And this is when two species are able to have progeny, but those offspring, the progeny, do not develop normally and have high mortality rates. A pretty good example of this is a liger. When a lion and a tiger breed with each other, they can produce an offspring that we call a liger. Now, often the offspring won't even make it to birth, but if it does, it has many health complications and often doesn't survive as long. And so they are reproductively isolated, lions and tigers, because their offspring are not as fit. There's also tigons, which is the opposite, when the opposite male or female of a lion and tiger mate with each other. And then the last one here is hybrid sterility. And this is when two species do produce an offspring, but that offspring cannot reproduce. And sort of a classic example of hybrid sterility is when you have a male donkey mate with a female horse, and they produce a mule. Now, this mule is healthy, however, it cannot reproduce. It is sterile. And so this is a reproductive isolating mechanism because the progeny that is produced cannot pass on those genes to the next generation. Okay, that is all I have for today's material. If you have any questions about it, please make sure you let me know so I can address any concerns or problems you might have. If not, I will see you in class. So bye for now.